Hi, my name is Lauren Quackenbush, and I'll be presenting my master's thesis proposal entitled Grapevine Rootstock and Cultivar Influence on the Rhizosphere Soil Microbiome. My thesis revolves around three main research topics: the grapevine rhizosphere microbiome, rootstock cultivar interactions, and genomic data science. This image highlights the rootstock cultivar interaction to point out some of the different considerations that go into rootstock and cultivar selection. Rootstocks are often chosen for their resistance to pests like phylloxera or nematode, or for their tolerance of certain soil conditions like salts or drought, while cultivars are often chosen for their phenology or winemaking characteristics. This research aims to illuminate another property that is unique to both rootstocks and cultivars, and that is their microbiomes. The purpose of this research is to articulate the core microbiomes of two different grape rootstocks and to identify indicator taxa of 4 different grape cultivars. The soil microbiome plays important roles in plant stress response, disease resistance, nutrient uptake, and carbon sequestration. So identifying the core microbiomes of specific rootstocks and cultivars would allow growers, viticulturists, and ecologists to better respond to both site-specific concerns like mineral deficiencies, suboptimal pH, and soil pathogens, as well as global concerns like carbon pollution. Both rootstocks and cultivars exert an influence on the grapevine microbiome, which has above and below ground components, illustrated by the matrix here, which shows differences in microbiome composition between different plant parts. The bulk soil and rhizosphere microbiomes are similar, but differ substantially from the above ground grape, leaf, and flower microbiomes. These unique assemblages are known to create unique expressions of microbial terroir. The reason why bulk soil and rhizosphere soil share similar microbial compositions is because they share primary controlling factors: the vineyard, year of collection, and soil temperature. The factors controlling microbiome diversity, though, are different and include soil pH, carbon-nitrogen ratio, total soil carbon, and soil moisture. Within the rhizosphere and roots specifically, the rootstock genotype and cultivar genotype exert a strong influence on microbiome diversity. Root exudates are known to influence the microbiome, and they include carbohydrates, amino acids, organic acids, and phenolics, which serve as a source of carbon and energy for microbes. Since the rootstock creates the root exudate molecules, this is one mechanism of rootstock microbiome influence. Rootstock influence on the microbiome is most significant in measures of alpha diversity. The table here shows alpha diversity measurements in the root and rhizosphere for 5 different rootstocks. As we can see, rootstock 420A in the middle has significantly lower richness, which is measured in operational taxonomic units or OTUs, and slightly lower Shannon diversity values in both the root and rhizosphere. The interaction between rootstock and cultivar genotypes results in a two-way influence. The cultivar exerts influence on the rhizosphere microbiome by regulating vascular uptake through stomatal conductance and by sending regulating signal molecules to the roots. The cultivar influence is measurable, but even more significant is the impact from the rootstock cultivar combination. As you can see in the histograms here, which compare the alpha diversity metric Faith's phylogenetic diversity, or Faith's PD, from the perspective of rootstock cultivar and rootstock cultivar interactions on the bottom here. So this bottom set of histograms really highlights how different microbial diversity can be even on the same rootstock when they're grown with different cultivars. So let's back up for a second and discuss diversity metrics. First of all, the metrics I chose to include were those that appeared the most often in scientific journal articles reporting on the same topics. There are 2 main categories of diversity used to describe microbiomes, and those are alpha diversity, which is used to measure variation within a sample type, and beta diversity, which is used to measure variation between different sample types. The alpha diversity metrics I'm including in my research are ASV richness, Pylus evenness, Shannon diversity, and Faith's PD. The beta diversity metrics I'm including are Bray-Curtis, weighted unifrac, and unweighted unifrac. The beta diversity results will be given dissimilarity distance coordinates and then plotted in a principal coordinate analysis matrix like the ones you see on this slide. Using these plotted graphs, I can make some assumptions about genotype selection based on groupings of coordinates. For example, in the above right graph, similar microbiomes are grouped together based on whether they were sampled from the bulk soil, rhizosphere, or root. So where does the data come from for the microbiome analysis? The answer is rRNA. Metagenomic sequencing refers to identifying the entire genetic profile of a sample, like a group photo. High-throughput amplicon sequencing is one processing method by which microbiomes can be studied. There are two main approaches to this kind of sequencing. The first is a whole genome shotgun approach, and the second is a marker gene approach. My research is using the latter, looking at specific rRNA component genes. The 16S rRNA gene is a widely used gene marker for bacterial and archaeal identification to the genus and species level. And the ITS gene is considered a universal fungi barcode marker. Once the genetic data has been captured with sequencing, it can be filtered, sorted, and identified using bioinformatics. Different genetic classification databases exist, and in studies of the Grapevine soil microbiome, the Ribosomal Database Project Classifier is one of the most widely used. I've identified three primary objectives to this research, and those are: one, to identify the core microbiomes of two grapevine rootstocks, 420A and 10114, and to identify indicator taxa of four Pinot Noir cultivars, Calera, 828, 777, and Mount Eden. Two, to determine the following metrics of alpha and beta diversity: ASV richness, Pylus evenness, Shannon diversity, Faith's PD, Bray-Curtis, weighted unifrac, and unweighted unifrac. Finally, to evaluate the relative influence of the following factors on the rhizosphere microbiome: rootstock cultivar interaction, pH, soil moisture, soil temperature, total carbon, total nitrogen, and carbon to nitrogen ratio. These objectives will serve to prove or negate my hypotheses that: one, the core microbiome of a rootstock can be better articulated by comparing the microbiomes of multiple cultivars grown on that rootstock, and two, that in the rhizosphere, the dominant influences shaping microbiome composition are edaphic, while the dominant influences shaping microbiome diversity are genotypic from rootstock, cultivar, or their combination. The experimental site is located in Santa Rosa, California, in Sonoma County. The four experimental Pinot Noir blocks are indicated on the map with pink stars. The two blocks on the right are on rootstock 10114, and the two blocks on the left are on rootstock 420A. I'll take 3 rhizosphere soil samples per block for a total of 12 samples. Each sample will be split into 2 bags, and one set of samples will be overnighted to Biome Makers Inc. in Davis, California for gene sequencing, while the other set of samples will travel with me to Cal Poly State University, San Luis Obispo, where I will perform soil chemical analysis at their lab under the supervision of Dr. Stuart Wilson. The sequencing results from Biome Makers will be sent to me as a file of paired end reads, which I can then use for bioinformatic analysis. The analysis of results will take several months, as I first use coding and bioinformatics software to filter, sort, and classify the samples. Operating in the QIIME 2 platform and incorporating multiple software packages, I will create both a phylogenetic tree and a rarefied species table that will be used as inputs for all of the alpha and beta diversity analyses. The species table is called the ASV table, which stands for amplicon sequence variants and refers to individual variations within the marker gene. Both ASVs and OTUs, which I mentioned earlier, are comparable taxonomic identifiers, and the main difference between them is just the version of software used. Following bioinformatic analysis, I'll use computational statistics to calculate diversity metrics and to test for significant differences between samples. Important for my research is that I will use a statistical approach to evaluate which variables are the most influential in shaping the rhizosphere microbiome. I've already taken some preliminary soil tests, both nutrient and microbial. The soil chemistry tests taken in March 2019 were broad composite samples that combined 5 blocks per sample, as you can see in the results table here. The microbiome tests taken in October of 2022 compared the rhizosphere microbiome to the bulk soil microbiome in 2 blocks, one on rootstock 420A and one on rootstock 10114. These fall samples will be incorporated into my thesis report for comparison, and in the case of the bulk soil results, as a control sample. These gene sequencing samples cost $250 a piece, and the price was split between me and Deloach Vineyards. This July, I'll take my final samples. These are the 12 samples from the 4 blocks that I discussed earlier. The reason I'm sampling in July is because, as illustrated by this graph, grape root production peaks at this time. Peak root growth is an important indicator of both nutrient uptake and microbial activity. So by sampling during a period of high nutrient uptake and microbial activity, when the leaf canopy area is high, I am hoping to capture a profile of the microbiome when the cultivar's influence is most pronounced. Costs for this sampling include the sequencing, which for 12 samples totals $3,000, and the travel and lodging expenses to and from Cal Poly, which should be around $400. These costs will be split between myself, Deloach Vineyards, and 1 or more educational grants. The soil chemical analyses don't have a laboratory cost because the lab and supervision are being provided as part of an educational exchange with Cal Poly San Luis Obispo. Once I have the results from both sequencing and soil testing, I can begin on the bioinformatic and statistical analysis of the data. In the meantime, I'll be working on my literature review. Looking ahead, I'm planning to have my final thesis paper complete by March 2024 so that I can prepare my thesis defense ahead of graduation in May of that year. I appreciate you listening to my presentation and welcome your comments and suggestions. Thank you so much.